So
You're not cognitively engaged.
I'm not cognitively engaged.
Mm-hmm.
So what I'm saying now is that with memory, is that it's responsible for converting short-term memory to long-term memory. Your long-term memory is stored in your brain, in the rest of your brain. So when we do experiments with mice and rats and we actually knock out a section of their brain, that doesn't necessarily affect memory in predictable ways. We would think that, okay, we just taught this rat something that maybe they'll forget a specific thing if we take out this region of their brain. No, it doesn't work like that. It matters how big of a chunk we take out. It doesn't matter where we take it out.
Interesting.
And what does that imply? That implies to us that memory is stored throughout the brain. How is that possible? It completely defeats me just telling you that the brain operates like a machine with different parts that do different functions. Again, adding to this beautiful mystery, adding to this complexity of this organ. Okay, moving on. But again, like I just said, how do we know what the different parts do? I want to introduce you to this guy. This guy here is named Phineas Gage, and he's not the most pleasant looking fellow you've ever seen. So let's talk about his story of what happened to Phineas. Phineas was on the railroad in the 1800s. He was the leader of his scouts that were working out there. And to build these railroads, it involved packing some dynamite down into these small holes that you put the railroad on. And I'm clearly not an expert on this, using metal rods. Okay, now one day Phineas was just going along, packing down, and a small explosion happened. And the metal rod ended up about 50 feet away.
Wow.
In this process, that metal rod went through Phineas's prefrontal cortex. Okay? So as you can see, it kind of went up his cheek, in the back of his eye, and through the front of his brain. Now, Phineas, after this, was not the same dude. Okay? He was a lot different. He was no longer the leader. He was no longer well respected. He was no longer liked. He was impulsive. His emotions were out of control. He was angry. He was violent. But what did this tell us about the brain? It told us that the front of our brain is probably involved in emotional regulation. It's probably involved in our impulsions. It's probably involved in the character flaws that Phineas demonstrated. So this is how science works right now. Science works by us, a lot of the times, knocking out different portions of the brain and seeing what happens. And this is early science at work. This is how we learn things. We often see people with certain disorders, people who have accidents, and we're able to see what they lose, and we're able to understand more about how the brain works.
So are there multiple people who've had accidents that were similar, where they could look at it and see if someone was had the same damage, that they show the same effects?
Yeah, I mean, that's what we do, is we see correlations. We see correlations between damage somewhere in the brain, and then we see behavioral outcomes that occur. Now, in science, we often say correlation does not mean causation. We have to show that this happens over and over and over and over and over again for us to draw that relationship. But yes, multiple people have had damage to the front of their head and have exhibited similar personality traits that Phineas showed. So let's look at some other forebrain structures here. We can look at, again, the thalamus. The thalamus is this relay center of the brain that you'll learn about in the introductory neuroscience lectures. Again, hippocampus, conversion of memory from short-term to long-term. The amygdala. This is one of those regions where really it does have a specific function. It is involved with fear. It is, if you again are to ablate the amygdala, you are to take out the amygdala in mice, they lose their sense of fear. You can go up to them with like a giant hot poker and just poke them. And they're like, it's fun. They don't even remember that, they're not even afraid of you. You could, if you find some like animal that's extremely afraid of humans or afraid of predation, and you take out their amygdala, they will not be afraid.
Wow.
That fear is pretty much gone. And then we have the basal ganglia, which is involved with motor learning and control. So again, looking at the basal ganglia, that's involved with motor control and learning. And look, I see I have a little statement here that says where memory is stored. Already covered that one.
All over the brain.
All over the brain. Okay. So again, looking at the hindbrain substructures, we have the cerebellum, which is involved with the coordination of motor control, and the medulla oblongata, again from the famous Adam Sandler movie, heart rate, respiration. Like we said before, I don't want you guys to memorize this stuff in detail. What I just want you to know is that the back of your brain, that's where vital functions are happening, okay? And that's what this is about. So, let's get to the neurons already. Let's get to the molecular basis of neuroscience. Just skipping a few slides here. So, we are going to take a trip into the brain. That's pretty cool, right? Zooms right in. So, we have the ultra-famous typical structure in the brain called a neuron. Now, let's imagine a neuron like a tree, okay? Let's imagine that the tree is receiving signals from its branches, and it is going down, and the signal is transmitting through its trunk and out its roots. Now, we can call the branches dendrites, we can call the trunk, we can call that the axon, and we can call the roots the terminal buttons, the axon terminals. Now, what we want to say is that the cell body here is where a lot of the neurotransmitters are down in the hillock, down in the bottom part of the cell body. A lot of the neurotransmitters are synthesized. And we have what are called action potentials, which we're going to explain in a minute, that are transmitted down the axon and into other neurons. We have the myelin sheath. The myelin sheath is kind of like a wire cover. It's helping to hold positive charge in. Did I mention that our brain operates on electricity? It's really how our brain works. It's a little misleading to just say, because everyone kind of knows that our brain works on electricity, but it really does work on this disproportion of charges on either side of a membrane. And that's why, again, we spent all this time looking at membranes, looking at biochemistry, looking at this stuff. So let's see what we got here on the next slide. Oh my word, what is that? That is a real picture of a neuron right there. That is a neuron that's been dyed with green fluorescent protein.
Wow.
And we talked about green fluorescent protein, that we actually take green fluorescent protein from a jellyfish, and we can see the structure of green fluorescent protein, and you all remember that proteins are made of amino acids, because you learned that. And this is what's called a beta barrel, okay? And we can see here that tyrosine 66 binds to glycine 67, and that forms, okay, just kidding. We're seeing that we form what's called a conjugated chromophore, okay? I don't want you to memorize that. All I want you to know is that this protein has a special way that it bonds in the middle of its center that causes green light to be emitted from it when we shine light on it. It basically emits green light. So what does that allow us to do? It allows us to see stuff. It allows us to see the neurons that we can stain. We can actually control which neurons are expressing green fluorescent protein.
Oh, wow.
Yeah. So, and this is a fun slide here, looking at this petri dish that a scientist/artist had gotten a colony of E. coli and got them to express some green fluorescent protein and red fluorescent protein, blue fluorescent protein, cyan fluorescent protein, into this beautiful beach scene of E. coli. So just again, there are amazing things that we do that you don't even think about, that are just important to know about the brain.